First, I'd like to thank the organisers for this wonderful opportunity to present my recent work. So I've recently joined the University of Exeter in the southwest of England after 3 years at MVBI. And the research I'll be presenting today was largely done during my time at MVBI and funded by Wellcome. Yeah. Okay, so we've heard a lot today about different ways, different aspects of the puzzle of classifying variants of unknown significance. And today I'm going to talk about one piece of the puzzle, which is functional assays. And since I got really interested in developing bioinformatics pipeline for new experiments, I'm going to talk about one of these, where we tried to create a prospective map of the effects of mutation in a disease. And so, you can just look at different variants that can occur in patients. Lots of them, as we heard today, are rare. Many of them might even be private, so you might want to have a database of variants of prospective variants that you can use when a patient comes up with a variant of unknown significance. And in addition to kind of this degree of pathogenicity, we also wanted to know more about how the mutation impacts the transcriptome as a whole in order to understand more, for example, how the mutation would avoid a drug. We also wanted this to be flexible, for example, to be able to use it in a drug screen to test drug resistance and how different mutations lead to drug resistance. So for that, we developed a single cell experimental and bioinformatics protocol, and we had a big pool of cells. Basically, we edited them so that each of them has individual variants of unknown significance, and then what we want to get at the end is both the phenotype across the whole transcriptome, so all the gene expressions for each individual cell, are also knowing the variant of the cell. And this was in collaboration with two great experimental colleagues from the Sanger Institute, Sarah Cooper and Matt Coelho. And so what they did was develop a way in which we can use CRISPR base editing. So this is a way of changing basically every C to T in a very specific window of 4 bases. And additionally, within this construct, so this is a construct in a virus that gets integrated in the DNA, similar to any virus that integrates in the DNA. Which means that this construct actually gets transcribed. And then this construct also contained 2 barcodes in different parts of the construct. And we use this in colon cancer cell line HT29, which has a specific BRAF variant. So then in order to link the whole transcriptome symbol cell information to the genotype, we used a way of editing those cells that we could turn off. So basically, these cells got this vector, and within this vector, they had these 2 barcodes. So one of them 12 nucleotides, the other 6 nucleotides, and these are all completely random. So basically, you have 18 nucleotides. It's very unlikely that 2 cells would have the same barcode. And so each cell has this individual barcode, and each cell has the edit. Then you turn off the editing, so the variant stays the same in the cell, but the cells grow. So you have little clones of cells indicated by the same color with the same genotype and the same barcode. Which then means we could take some of them to do transcriptomics and some of them to do DNA sequencing. And since these barcodes get transcribed, we can find them in the RNA. And we can also find them in the DNA by using amplicon sequencing with 39 amplicons that spanned a gene of interest that we looked at, which in this case was JAK1, and 2 additional amplicons across the 2 barcodes and a so-called guide RNA. And so from a bioinformatics perspective, so many of you might have worked with single cell and just seen, okay, this is quite noisy data, because obviously we have information only from a single cell, so across all transcriptomes, it tends to be quite sparse. And in addition to that, both on the RNA and the DNA side, we use droplet-based methods, which means that you have, the material that gets sequenced is from a droplet that contains, well, most of them don't even contain a cell. Basically, it doesn't contain a cell. It contains cell-specific DNA or RNA, and then it contains something called ambient DNA or RNA, which is just material from cells floating around, from dead cells floating around. So in effect, you can then basically just map your reads for these barcode amplicons, just the same way as you would just map any DNA or RNA sequencing. And you can count each of the barcodes. And you get basically a matrix for each cell and the different barcode counts. And you get it on both the DNA and the RNA side. And then you see here that in many cases, it's just very hard to decide, are you going to say this cell has the barcode or not? And we found that this is an extremely important part of the analysis, because if you get this wrong, then you just don't find any signal, because you have cells with a random genotype. And yeah. So then in order to sort this out, I looked at the distribution that these different barcodes follow. And so basically you have these smaller counts that are like noise. So you have this cluster here on the left that's it is skewed. And then on the right, you also have a skewed extra bin, basically a skewed bin on the right. And they also found that for RNA, it tends to be 2 bins. So there's an extra small peak, which probably is because this is transcribed, and so how much this gets transcribed depends on where exactly it happened to get integrated in the DNA, so whether this was a more or less active region. And so you see this is still pretty noisy. I mean, if you drew a line, where exactly would you draw the line for these clusters? And obviously we can use an algorithm to do that, but still uncertain. So in order to avoid problems, because of that, I said that a barcode is in a cell only if it has at least a 90% probability in being one of the higher clusters. And then I say there's no other barcode has a probability of less than 10% in being there. So this avoids calling those that might have multiple barcodes or not, or you don't know which one it has. So then this allowed us to quite confidently call those different barcodes. And so when we designed the experiment, we decided to go for HT29 cells, because that was a cell that we knew very well, and we had done other work on it before. And what we hadn't thought about is there's triploid cells, and we're looking at single cells, and we tried to genotype them. So we found that actually our barcode strategy is also very important in getting the right genotype, because what you find is this, in addition to this ambient noise, you have all kinds of small counts for other variants in your droplet. And then you also have something called allele dropout. So basically, you maybe see only 2 alleles out of the 3. And so you see that if you just use standard variance calling, you would get lots and lots of mutations just on one allele that are not actually true, because then if we if we check consistency across the barcodes, we actually see that that most cells do not have variants that are just in one allele. So basically, otherwise, we would find that each cell has on average four different mutations just in one allele, and that's just not true. So this leads me to telling you why actually we had to do all this complicated stuff in a way. So you could just look at this so-called guide RNA. So that's basically a sequence that tells you where you're going to cut the DNA and edit it. And then theoretically, so this is different for like different experiments. And in ours, this is set up so that it should edit between base 5 and 9 of this guide RNA, where this guide RNA binds. And we see that that's many say on this figure on the left, you see the 2 bars is where it should edit, basically. And you see that most of the homozygous mutations are actually in that area, but some of them are actually earlier on. So we're missing some of them just by predicting. And when we look at basically whether when we only look at homozygous mutations and see whether, so does each cell basically have the mutation that was predicted, and we found that exactly half of them, funnily, it's exactly 50% here, had the edit exactly as predicted, which isn't actually bad, but it's just good to know which exact mutation you should have. Okay, so that's the genotype. And now, obviously, the reason why we're doing this is because we want to see how the genotype is related to the phenotype. And in this case, it was, so we looked at JAK1 mutations in HC29 cells and looked at interferon gamma response. So we had a very strong phenotype. And so you can see we have very, very strongly, so when we look at 2 clusters, one that consists of both wild type and edited cells, and the other that consists of only edited cells, then we see that between those 2 clusters, we see a strong effect on the JAK-STAT pathway. And so these two clusters you see here, there's this right one where there are basically almost all the wild type cells are there. So almost all the cells where there was no edit. And then there are also lots of edited ones that just didn't have an effect. And then there's another cluster with lots of edited ones. And when we then look in more detail at how they are, what they look like across the whole transcriptome, we see that these are very correlated, okay, which is expected because we're just mutating this one gene. And so then we wanted, in addition to this very rich phenotype, we actually wanted to get a score similar to a score from a survival screen or so, in order basically to just assess the level of malignancy of each mutation. So we want to basically something that has wild type and synonymous mutations on one and subclonals on the other. And it's based only on the transcriptome. And then we decided to use Laplacian eigenmaps, also called diffusion maps. So that's a way to take your transcriptome and put it into 2 dimensions, basically you see the strongest. So it's a nonlinear way of getting the strongest axis of variation. And because it has such a strong effect, this works really well here. And in fact, we only looked at the first dimension of this and saw that stop codons are very nicely on one side and wild type and synonymous very nicely on the other. And the missense mutations are on both sides and also a little bit in between. Now, this score that was based on the transcriptome then allowed us to go back and say, okay, does actually our experimental bioinformatics pipeline give right results? So can we look at like mutations that we definitely know, for example, stop, do they really have a very high diffusion score? We saw that, so doing this, our method, we got a precision recall of 96%. And if we had only done the predictions, the precision would go down to 78%. And now, okay, do we really need to care about knowing every edit exactly? And the answer is yes, we do, because in our case, variants are really only effective if they were all 3 alleles. So if there are only 2 of them, then there's no effect here. Similarly, a change of just 1 base in where the homozygous mutation can come from totally benign to very deleterious. And in addition, looking at these diffusion scores and looking at each individual gene on the level of these diffusion scores allowed us to find some very interesting variants. So here on the right, you see the distribution of these diffusion scores across each individual cell. So that's why it's a distribution. And you see these benign ones on the left, and then we have pathogenic ones. And then we found those interesting ones in the middle that were like all over the place. And then we went back to our previous data and realized that these are what we previously called separation of function variants, which had a very, very strong proliferation effect, stronger than some of these variants on the very right here, but a lower effect on the JAK-STAT pathway. Okay, so this basically has allowed us to test our method, make sure it works, and then we wanted to use it in several applications. And one of them was drug resistance. So this is again HG29 cell line, and in this case here we can use base editing to look at different variants and edit a combination drug treatment. We were interested in understanding the difference in terms of transcriptome between canonical drug resistance and drug addiction. And drug addiction means, so it's this brown cluster on the right. So this means that if the drug isn't there, it actually they grow less. So it would actually be good. But if the drug is there, they grow more. So actually, if a patient has that, you might consider a drug holiday. It's important to understand what they do. And again, basically, we looked at both the rich phenotype, also found new mutations not previously associated with other drug resistance. And again, we found a very correlated group of these strong effect mutations that were mostly drug addiction variants. And so this score again allowed us also to see that these unfortunately are very correlated with poor patient outcomes. And yeah, so currently we are exploring further applications of this. And I would like to thank my collaborators, especially Sarah, Matt, and also Andrew Bassett from the Sanger Institute. And I would like to thank Open Targets for funding some of the experiments, and my postdoc supervisor, John Marioni, and the Wellcome Trust for funding my salary, allowing me to come here and do all this cool research. Okay, thank you.
Time for one question while Ning-Chen, you want to come and set up your talk? Yes, can you perhaps tell us a bit more about these pathways that are typical for the resistance?
So here we looked specifically about MAP kinase, because one of the drugs we use is also MAP kinase inhibitor. And so there's also, so the cell line also has a BRAF V600 mutation. So the other drug we used is BRAF inhibition.
Thanks. Other questions? Okay, then. Thank you, Magdalena.